Hello biologists, this podcast today is on plasma membranes. Hopefully you're looking at this structure and you're going, alright, this is made up of a phospholipid bilayer and we've got our heads and our tails. Obviously the phospholipids are arranged like that for a reason, so we're going to go through that in this little podcast today, and we're also going to go through the process of diffusion, so the movement of substances through this membrane. We've been talking about in class about all cells and the common things that you find between cells being prokaryotic or eukaryotic. One of those things is that they all have a plasma membrane or a cell membrane. So hopefully you're thinking, why do they need to have this membrane? Obviously, it's really important for a cell to sort of control what goes in and out, and so that's what the plasma membrane does. If we look at the membrane here, it's composed of a double layer, so what we call a bilayer of phospholipids. Now we've learnt about lipids in class already, but this is what the phospholipid bilayer looks like. Okay, so let's have a closer look at those phospholipids. So phospholipids, we call them amphipathic lipids because they have a hydrophilic head which is composed of phosphate, among other things, and it has hydrophobic tail made of two fatty acid chains. So it looks like that. So we've got our head, which is hydrophilic. So we like to say that has a high affinity to water rather than water loving. And it has these two fatty acid tails or chains, which are hydrophobic. So they actually repel water. Because these phospholipids have both a hydrophilic and hydrophobic region, we call them amphipathic. So it's really important because often when we talk about lipids, we say that the majority of them are hydrophobic, but that's obviously not the case with phospholipids. We also can call the head and tail polar and non-polar. So you might see this term, or actually you'll definitely see this term pop up, especially when we talk about diffusion. Polar means it's charged, and then the tails are non-polar. So they're not charged. Another way of remembering it is anything that's hydrophilic is polar, and anything that is hydrophobic is nonpolar. So this is what the plasma membrane looks like, and in class we would have called this the fluid mosaic model, the common model that we sort of use to explain the structure of the membrane. So it's really important that you draw this right if you're asked to draw it. Particularly, the examiners are going to look for the phospholipids, that all the tails, so these tails here are facing each other, all right? And you'll also notice that we've got some of these other things embedded within the membrane. So we've got some transport proteins. We've also got these carbohydrate chains, which, well, in this case is attached to a protein here, okay? So basically, the function of this phospholipid bilayer is to exhibit selective permeability. So that means that only certain substances can move through the phospholipid bilayer, and other substances may have to use these proteins here. So things that are non-polar, such as oxygen or carbon dioxide, can easily move through the phospholipid bilayer, as I've shown there, so can easily move through. But polar substances, things like water, glucose, ions, have a really tough time diffusing through this phospholipid bilayer. It wouldn't happen very readily. So what they need are these proteins that are embedded in the membrane just there. The carbohydrate chains here, you might remember from your holiday homework, they aid in cell-to-cell recognition. And one other structure here that isn't drawn is the cholesterol, which stabilizes the plasma membrane. So you find the cholesterol between the tails in here, just sort of throughout the membrane, and basically they allow the membrane to be as stable as possible, so fluid enough that things can move through easily enough, but also stable enough so that it doesn't break. So we're going to look at the processes of diffusion. There's a couple of types we're going to learn today, depending on the type of molecule that we're looking at. So simple diffusion is when we're going with the concentration gradient. Now, this is a term that you're going to see quite a bit. Going with the concentration gradient means it's going from an area of high to an area of low concentration, which, as you can imagine, would be quite easy. Because of that, it's passive, so it means that it doesn't need energy to actually happen. And obviously, the larger the concentration gradient, so the larger the difference, the more rapid the rate of diffusion is going to be. So this diagram here shows you the simple diffusion of a substance through the phospholipid bilayer. So this could be oxygen, all right? So it's moving from an area of high concentration to an area of low concentration. Another type of diffusion is facilitated. Now, in this case, the molecules may be larger or they might be polar, so they have trouble getting through the phospholipid bilayer. And what they do is they actually they go through the membranes that were, the proteins that we're seeing embedded in the membranes, and there's two types. There's the channel proteins, which basically provide corridors, and also there's the carrier proteins, which sort of undergo a little change, and they basically bind to and assist the molecule through the protein. And it's also important to note that each protein is designed to recognize only one or a group of similar substances. So depending on what we're looking at, there's going to be a few different ones. So things like glucose would do this, right? So amino acids, another one. So again, they're going from, you will notice here, from an area of high concentration to an area of low concentration. So it's passive, right? This is showing you the channel one, so it's just a corridor. But this one is showing you how it can act as a carrier, so where it actually changes shape subtly just to assist its movement through. So it's really important to note that facilitated diffusion is still passive. If we look now at active transport is basically they're acting as pumps, and the proteins that we looked at before, they pump these substances across or against their concentration gradient. So this means that they're moving from an area of low concentration to an area of high concentration. And to be able to do this, you need ATP, or energy. So that's adenosine triphosphate. For those who don't know, we're going to learn a bit about that a bit later. And this is really important as well. So things like maintaining a lower concentration of sodium ions, which is you're going to see that pop up a bit later in the course when we learn about neurons and how, you know, those things pass through the neural pathways. So that's really important there. And so this is just a diagram showing you that process. So as I said, area of high concentration to an area of low concentration. So that's normally what happens with passive transport. But as you can see here with active, it's going against that concentration gradient. So energy is required to pump, usually ions, something like that, through the membrane there against its concentration gradient. Another type of diffusion that we're going to talk about is osmosis, and that's the net movement of free water molecules from an area of where it's quite dilute to an area where it's quite concentrated. Okay, so I'll show you a picture of that in a minute. It's passive still because we're going with the concentration gradient. And examples of this, so, you know, water osmoregulation in humans, so making sure that we have enough water in our bodies, our cells are hydrated enough, and then, you know, getting rid of excess water through the kidneys. So this is a picture showing you what happens in osmosis. So hypotonic, hypertonic, these are terms that came up in your holiday homework. Basically, hypotonic means that the solution isn't as concentrated as a hypertonic, all right? So as you can see here, it's really important we always have a selectively permeable membrane. So that means that water can move through, but not these ions, whatever they are, or solute, I should say. So it could be salt or sugar. So this one is less concentrated. This one is high concentration. So what's going to happen, all the free water molecules in here are going to want to move through to the area where there's a higher solute concentration. All right, so the water is going to move through there. And as you can see then, in this particular instrument, you're going to get different levels of water on either side. So this side, the level's going to drop because the water has moved through to that side because there's a higher solute concentration. Really important when you're defining osmosis that you talk about the movement of free water molecules from an area of low solute concentration to an area of high solute concentration. Just be really careful how you define it. So in summary, it was only a short podcast. The phospholipid bilayer allows the membrane to be selectively permeable, and that's obviously very important in terms of how the cell functions. Non-polar molecules such as oxygen, carbon dioxide, readily diffuse through the phospholipid bilayer, but polar molecules do not, so things like glucose, amino acids, and they instead rely on proteins that are embedded in the membrane. Some methods of diffusion move molecules with the concentration gradient and are passive, so that's simple, facilitated, and osmosis. But some methods require ATP, so adenosine triphosphate, to actively pump these molecules against their concentration gradient. So I hope that was enough today just to give you an idea about the phospholipid bilayer and how membranes work and the process of diffusion. We'll obviously be discussing this a lot more in class. Any questions, then just email me. Thanks, guys. Bye.